All right, so these were the steps we just went through, and let's actually just review it one more time by going through this these images. So first thing is we have calcium coming in from the outside environment as well as from the ER where it's being stored. Okay, it's going to activate this calcium channel modulating complex, and the activation of this complex turns on MLCK, this enzyme, which will then either proceed to phosphorylate the myosin head and bring about muscle contraction, or in the case of muscle relaxation, the phosphatases will take over and you will cease muscle contraction, right? That's the opposite there. Okay, important to understand as well is that calcium must be removed from the cytoplasm. This is something we saw with skeletal muscle as well. So the presence of calcium ATPase pumps, as well as the calcium sodium transporter or countertransporter, are important for getting rid of that excess calcium in the cytoplasm. Right? The muscle will not cease contraction until the calcium has left the cytoplasm. And so these two methods of transport remove some of that excess calcium. Okay. And also worth mentioning here again is that the rate of this muscle contraction, or the speed rather, is much slower than what we see with skeletal muscle, about 10 to 100 times slower. Again, because of that myosin head moving all the way across the actin filament, as well as the smaller cell and this muscle not being optimized for speed the way skeletal muscle is. Okay. Now let's talk about the neuroregulation of smooth muscle. We know that the autonomic nervous system is innervating smooth muscle, so there's the parasympathetic division and the sympathetic division. You guys should be experts in that by now, right? We know that as a result of this, we can either excite smooth muscle or inhibit it. Very opposite from what we saw with skeletal muscle. So we can only excite skeletal muscle. Here, not so much. We can excite it or inhibit it, depending on which division of the autonomic nervous system is being is innervating or is dominant. So the precise response depends on the specific receptor. We looked at those adrenergic receptors. They had different locations and different functions, and so we know this, right? The exact response we get will depend on the receptor type and its second messenger pathway and its activity on the tissue. We also talked about neurotransmitters being released from varicosities here. Okay, so this differs greatly from the discrete one-on-one neuron communicating with each other. And instead, we have these swellings along the nerve terminal in these neurons, and then these neurotransmitters are released from these swellings, and the swellings we know are varicosities. Okay, and then the neurotransmitter diffuse and bind to various receptors on the affected tissue. Okay, so this slide is really a review of things we already know about the neuroaffective junction, about the autonomic nervous system, and about the neurotransmitters that bring about the response here. Now, on that basis, we're going to speak about two types of smooth muscle: single unit smooth muscle and multi-unit smooth muscle. All right? And they basically differ in their structure, but then their function as well. So what type of tissue they're typically found in, how they contract, how they behave. Now, single unit smooth muscle is the most abundant type throughout the body. We see it in the intestinal tract, we see it lining blood vessels, as well as in the respiratory tract. And it's heavily reliant upon connection between each of the cells via gap junction. Can anyone remind us what a gap junction is, or what the main function of a gap junction is? We spoke about gap junctions already. So what's the function of a gap junction? Despite the name gap junction, is it holding two cells together? Is it like a anchoring junction? It's not, right? It's for communication. So a gap junction is like a hollow opening between two cells that is very optimized to allow cells to communicate. And so because of the large number of gap junctions between the single unit smooth muscle cells, we have this type of muscle contracting as a unit. Okay, so all the cells are on the same page, they're sharing information, they're synchronizing with one another. And so the contraction of this type of smooth muscle happens as a single unit, hence the term single unit smooth muscle. All right, so I really want you to make that connection between the presence of gap junctions, which allow for cell-to-cell communication, and the way that this type of smooth muscle behaves. Also important with single unit smooth muscle is that they have the ability to spontaneously depolarize. Okay, and this is tied into their ability to contract without stimulation. So in the absence of being stimulated, these cells can sort of spontaneously bring themselves or spontaneously change the membrane potential and depolarize. Okay, and this is what we call tone, that spontaneous depolarization without any direct stimulation. Now they can increase or decrease in the amount of tension that they display, as well as display what's called a stretch reflex. So there's a sudden stretch which induces relaxation of this muscle, kind of a reflex for being overexerted or being overstretched. Now, as a result of this type of tissue acting as a single unit, do we expect to see recruitment? So we know what recruitment is. Do we expect to see recruitment in this type of tissue? Would you expect to see recruitment in this type of tissue? No, I mean no. Why? Why not? It's all connected. So it's acting as a single unit. All of the cells are on the same page. They're communicating to each other via these gap junctions. And so there's no one left out to really recruit. Okay, and so recruitment is not seen with this type of muscle. And then lastly, there's varying amounts of intracellular calcium, which brings about varying degrees of tension produced there. When we talk about multi-unit smooth muscle, we're going to see the exact opposite property. So there are very few, if any, gap junctions present in this type of muscle. And so this type of muscle does not have synchronized action the way single unit does. And so each of these cells behaves independently or differently, hence the term multi-unit. We typically find this type of muscle in the large airways, in the large arteries, all right? And this is going to be important to correlate with the function. So in large airways and large arteries, we typically want lots of specialization, lots of personalization of the different parts of the muscle. And so in big areas like this, we will see multi-unit smooth muscle. Whereas smaller areas such as the vessels, parts of the respiratory tract, we don't need much variation, much personalization with the smooth muscle. And so we typically see that single unit type of muscle in those areas. Okay, so just to reiterate here, this type of fiber receives its own innervation. So each cell, right, very different from what we saw with single unit. Each cell is innervated independently. Each cell comes into contact with one of these varicosities, and so it acts as its own boss. There are very little gap junctions, and so each cell is regulating its own contraction. So as a result, we do not see tone, and we can see recruitment. So if 3 cells are contracting, we can go ahead and recruit a fourth cell, and a fifth cell, and a sixth cell. So because each of these cells acts independently, we can see recruitment in this type of tissue. And then lastly, we'll talk about pacemaker potential here. And this is going to segue or transition us nicely into the cardiovascular system, which is also a tissue that exhibits pacemaker potential. So pacemaker potential, again, is that spontaneous depolarization of a cell to threshold, or sometimes not quite to threshold. So we have 2 types. There are slow wave potentials, and these are basically changes in the resting membrane potential. Questions? So basically, we can spontaneously change the resting membrane potential here and depolarize this cell to threshold, which generates a pacemaker potential. Okay, so these are 2 types of pacemaker potentials that we see with smooth